Treatment for the blood cancer ALL, or acute lymphoblastic leukemia, typically involves the use of asparaginase, which is an enzyme or a reaction helper that breaks down asparagine. So, asparagine is an amino acid, it's a protein letter, and it turns out that for reasons that aren't really well known, in this cancer, in this ALL, these cells are making less of the asparagine maker, asparagine synthetase. And so they're relying on getting asparagine pre-made through the bloodstream. If you introduce into the bloodstream more asparaginase, which is the breaker, the asparagine breaker, then you're going to break down the asparagine that's in the bloodstream and then deprive those cancer cells of the asparagine that they need. Now, the healthy cells, they'll be fine because they still make enough of the asparagine synthetase. And so they're going to make enough of the asparagine to get by, even though you're depleting the asparagine that was made into the bloodstream. And so these cancer cells are going to not be able to compensate, however, so they're going to be selectively killed, whereas the healthy cells, they'll be fine because they can still be making more of the asparagine. And so by adding this enzyme by giving these patients more of this enzyme, so they normally make this enzyme, but we can also give it to them. Typically, we give them like the E. coli version or another bacterial strain if they develop resistant or if they develop like an immune reaction to the E. coli one, and I'll get into some of this. But you give them more of this enzyme, and then this is going to selectively kill the cancer cells. It can also, though, cause metabolic changes through the rest of the body because you're messing up some other things, so it's not without side effects, and there are problems with people developing like sensitivity as well as resistance, and so I'll get into some of these things as well as a broader overview of some of the history of this treatment. And then if you stick around, I'll get really into the geeky nitty-gritty of how this reaction mechanisms actually work with the synthetase and so the maker and the asparaginase, the breaker, because there's some pretty cool biochemistry going on, but I'll leave that to the end in case people wanted it earlier, but I hope you'll stick around. So let's dive in.
Let me just start by saying I'm a biochemist and not an oncologist. I'm the geek kind of doctor, so a PhD, and not the medical kind, not an MD. So if you have medical questions, please talk to a medical doctor, talk to your physician, but I'm just going to try to talk about the biochemistry behind this treatment. So first off, what is leukemia? Well, leukemia is an umbrella term for cancers that affect the blood and the bone marrow, which is where the blood cells are made. There are different types of leukemias, and they're typically named in terms of what type of blood cells that they affect. In the case of ALL, this acute lymphoblastic leukemia, they're affecting a type of blood cell called the lymphoid progenitor cells. These are a type of white blood cells, so these are a type of immune blood cells that are important for the immune system. And these are going to, if you have problems with these lymphoid progenitor cells, this is progenitor, it's like they're making more cells. And so if you have a problem with these progenitor cells, the cells that more cells are made from, you're going to have a lot of problems. And so ALL can cause problems, including anemia, fatigue, and immune system dysfunction. And in this disease is often characterized by having a decrease in the production of asparagine synthetase, which is needed to make asparagine. And this is the basis for this vulnerability, that we can target we can make them deficient in this by depleting the asparagine that's already made. So, and then they can't make more, and then they're going to be selectively killed. So asparagine, just a note about it. So this is what it looks like. And it's a building block of proteins. So it's one of the 20 common proteinogenic amino acids. So one of the 20 protein letters that can be linked up into chains to make proteins that can then go do important things throughout the cells. And so asparagine looks a lot like one of the other amino acids, aspartate, which we looked at in my post yesterday. And so you can see that they look really similar, except that asparagine, it has this amide group on the end. We'll talk more about this when I get to the really geeky part at the end. But it has this amide group. You have this nitrogen containing group with this carbonyl carbon, so the C double bonded to an oxygen. Here, you still have that carbonyl group, that C double bonded to an oxygen, but here it's bound to another oxygen, and we call this a carboxylate group. And sometimes you'll see it drawn in the carboxylic acid form, which is just its protonated form. But you can see that these look really similar. And in fact, you can actually convert between the two, and this is how you can make more asparagine from aspartate using asparagine synthetase. And so basically all you have to do is take the amino group, take an NH3 group from one of them, from something, and transfer it onto it. And in the case of asparagine and asparagine synthetase, we're going to be transferring an amino group from the amino acid glutamine to aspartate to make asparagine. Here you can see that we're changing two amino acids into two different amino acids. So we're taking aspartate and glutamine, and we're transferring the amino group from glutamine to aspartate to make asparagine, and leaving you with glutamate once you've taken that NH3, that nitrogen group, that amino group. And so this is what's going to happen with the help of asparagine synthetase. And so normally what happens is that the cells are going to secrete asparagine into the bloodstream. And so there's going to be plenty of asparagine into the bloodstream. And so your healthy cells, they're going to be able to, if they wanted, they can take some asparagine from the bloodstream, but they can also make their own asparagine from this aspartate using their asparagine synthetase in that reaction that we just looked at, and we'll look at in a lot more detail later. So they're able to make more of this asparagine. And but leukemic cells, they're not able to make the asparagine from the aspartate, or at least not much of it. So what's going to happen is that they're just going to take it out of the bloodstream. And this is going to be fine normally because there's plenty of asparagine in the bloodstream. But what happens is that if you add asparaginase, so if you add more of this breaker, so normally our cells make some of this asparaginase, but they control the levels that they make so that they can tightly regulate how much asparagine they have and how much aspartate they have. And if you add more of this asparaginase, well, what this asparaginase does is it kind of reverses this, but not in the same way. But it's going to remove that amino group. And so here you can see it's getting removed as ammonia rather than transferring it onto another amino acid. So instead of doing a transamination reaction, it's just doing a deamination reaction. And so it's just removing that ammonia group. It's deamination reaction. And we'll look at more in that in more detail later as well. But you can see that you're destroying the asparagine that you have. And so the healthy cells, they'll be able to compensate. So if you take the asparagine out of the bloodstream, they're going to have less of it in the bloodstream. And instead, they're going to have more aspartate in the bloodstream, which they can deal with. Then they can convert that aspartate into asparagine. And so they're able to make plenty of asparagine that they can then use. But in the case of the leukemic cells, well, here they don't have the ability to make the asparagine from the aspartate. And so they're going to get low on asparagine. And what's even worse is that because you're depleting it from the bloodstream, you're going to have these cells actually kind of like shipping out the asparagine that they do have into the bloodstream, so that there's plenty in the bloodstream. So you have these cells depleting the little that they do have. And so this is going to cause those cells to die. And this is the basis for this treatment. And so since they don't make much of the maker, and if you add more of the breaker, they can't compensate. And so this treatment was first tried out in the 1960s, and it's become a mainstay of treatment for childhood ALL. And it's typically given in addition to traditional chemotherapy drugs, things like vincristine and prednisone. It's also used to treat some forms of other cancers like acute myeloblastic leukemia, or AML. And so basically E. coli is an AS. So yes, they're taking this protein and purifying it out of E. coli. So out of bacteria, at least traditionally it's made from E. coli. And so this might sound really, really weird, but if you're into molecular biology, so if you are doing any sort of genetic cloning or things like this, where you're stitching together pieces of DNA or you're analyzing DNA, basically you'll be really, really used to using things that are made in bacteria, because bacteria make a lot of useful things, including like restriction enzymes, which recognize and cut specific DNA sequences. So we use things that were made from bacteria a lot. And in the case of asparaginase, it turns out that they make a lot of a good asparaginase, and so it can be purified and used as treatment. But it's not perfect. And so some people, what happens is that they get a hypersensitivity reaction to it. So basically they have a sort of like immune response to this E. coli derived asparaginase. What can be done is that one thing that can help is pegylation. So PEG is just like this sugary molecule, and it's often added to a lot of different drugs. What it does is it helps with solubility, and it also kind of hides it from the immune system. So this like oncaspar and asparagus are pegylated versions of the native E. coli asparaginase. And so basically native means we just purify the bacteria's own thing without modifying it, at least initially, and then we add on the pegylation in the case of these. But people still have immune reactions and hypersensitivity to the E. coli. And so there's also another strain of bacteria that can be used, Erwinia chrysanthemi. And basically, this is another bacterial strain. And because it's a different bacteria, people who have an allergic reaction to the E. coli one, well, now they might not have the same reaction to this other bacteria, asparaginase. And so Erwinase is the natural version of asparaginase. And what can happen though is, so here's a nice review article, and it turns out that this is going to, the problem with one of the problems with this is that there was like production problems and a shortage of this Erwinia chrysanthemi natural bacteria. And so people started turning to recombinant technology. So recombinant technology is when we can kind of stitch together pieces of DNA or recombine them. And what we can do is we can take the instructions for making the protein, take the instructions for making the asparaginase, and stick it into like a plasmid, so a circular piece of DNA that we can then stick in bacteria and get bacteria to make this for us. And so you might say, well, why do you want to do that if you already have the bacteria making their own? But here you can do things like change the sequence, and you can make any strain of like any version of it that you want, and you can make it in a strain of bacteria that's going to make it so that it's people will have less reactivity to it. And you can also then make a lot of it. So make the bacteria make more than they would normally make. And this is going to allow you to make, to like have better yields from the purification process and things. And so there's recombinant E. coli derived asparaginase spectrilla. And then recently there's this, recombinant Erwinia chrysanthemi, this rylase. And so you can learn more about that. I'll post links to these nice review articles and things like that. But so yes, so hopefully this will help clear up some of those problems that are caused by the sensitivity reactions, as well as by the not having enough of these enzymes. But there's still other problems because, well, it turns out that although those cells, the healthy cells, can still make the asparaginase, and so, or the asparagine synthetase, so they can still compensate and make more of the asparagine, it turns out that you're messing up other things too. You can have global changes in the metabolism. So metabolism is the making and the breaking of molecules. And we've been talking about the metabolism of asparagine, but it turns out that asparaginase isn't totally specific for asparagine. Asparaginase can also deaminate glutamine. So glutamine looks a lot like asparagine, but it has an extra carbon in its chain, and it can kind of like trick asparaginase into breaking it down. Well, not really trick it, but I mean, it has less. Asparaginase prefers to break down asparagine, but it can also break down glutamine. When you break down glutamine, this is a big deal because, well, you get glutamate, and glutamate is at the heart of a lot of different metabolic pathways, and it's responsible for a lot of things, and it's also a key regulatory molecule, helping control what types of metabolic reactions are going on. And so if you're messing with the levels of glutamate, then you're going to be causing global changes, things like relying less on glycolysis, less on sugar breakdown, and more on using fats and things like this. Basically, metabolism is not just single pathways, it's an interconnected web, kind of like a complex subway system, and everything is affecting everything else at some level, typically. And so this treatment can have global effects, and it's not just like you can exactly target those cancer cells. Another problem with this treatment is that cancer patients can develop resistance. So this is showing you, this is from a post I did on antibiotic resistance, but the same sort of thing is happening in the case of cancer treatment, where these cells are rapidly dividing. Cancers are characterized by having like uncontrolled growth. And so these cells are rapidly dividing and dividing and dividing. And each time they divide, they have to copy their DNA. And when they copy their DNA, they can have like random errors in the copying. And if those errors make it so that they can't grow or survive, then they're going to die out. But if those errors give them a growth advantage, they're going to survive. And if you're killing off most of the cells, but there's some cells that happen to have a mutation that makes it so that they can still survive in the absence, when you put more asparaginase, then they're going to be able to still survive in the presence of the treatment. And when everything else is dying, well, now they're going to be the lone survivors left. And so they're going to be able to multiply. And then you end up with a cell population that's resistant to the treatment. So the ways that some ways that the cells can do this is they can like upregulate the maker, so they make more of the synthetase, so that they're more like the normal cells, so they start making this again and making more of it, so that they can still survive. And so there's some treatment, looking into treatments with trying to add like an asparagine synthetase inhibitor to try to combat this. But then again, you're going to be having more risk of affecting the normal cells. Other ways that cancer cells can develop resistance is through like breaking down proteins instead. To get the asparagine that they need, just get it from breaking down proteins. And so there's some looking into like protease inhibitors and things like this to prevent those protein breakdown. In terms of, so that's the future of the treatment potentially, in terms of the history. So it goes back to like the 1950s and 60s. In 1953, kid found that guinea pig serum caused regression of implanted lymphoma cells in mice. So it was helping cure the mice. He wasn't really sure why. But then in follow-up studies, Broom found that this effect was coming from the asparaginase. And then in the 1960s, Delawi et al. And Hill et al., they tried this treatment in human patients, but there wasn't really that much of it. And it didn't really take off until the 1970s, when they could produce this at larger scale so that they had enough to do the large trials that they needed in order to show it was effective. But those trials did show it was effective, and so it's become a standard part of the childhood ALL treatment. It was approved for medical use in the US in 1978, and it's on the WHO's list of essential medicines. And I will link to more resources and helpful review articles and things like that in the comments in the blog post. And now I want to talk more about the mechanism. So we're going to be looking at the mechanisms of asparagine synthetase and asparaginase. First, I just want to make a note about asparagine. And you might be wondering, okay, why is asparagine not positively charged? Why is this nitrogen not acting as a base, taking a proton, becoming positively charged? And you might be wondering this because, well, the other day we looked at lysine, the amino acid lysine, and the lysine, it had a nitrogen on its end, and it was almost always protonated. It would act as a base. The neutral form acts as a base, takes a proton, and now you have this positively charged. So why don't we see this with lysine? Well, with lysine, what we have here is we have a primary amino group. We have a nitrogen that's just attached to a carbon. But what we have with asparagine is we have a nitrogen that this carbon that it's attached to is actually a carbonyl carbon. Instead of just having like a normal amino group, we have an amide group. And an amide group is going to act very, very differently because this amide group is what we call resonance stabilized. So basically all of these different atoms are connected by sharing pairs of electrons. And I have much, much more on this in other posts. But you share one pair for a single bond and two pairs for a double bond. But then you can also have this thing called resonance or electron delocalization, where you have electrons shared between like multiple atoms that aren't directly next to each other. And so what's going to happen is that you're going to have like more than enough for a single bond, but not enough for a double bond for both of these. And so you get kind of partial double bond character between them. And this is resonance delocalization, this resonance stabilization. This is going to make it really stable and make asparagine really happy as is with this amide group. And this is the same kind of bond we see in the peptide backbone that we've talked about in other posts. But basically, this is going to be very, very happy. And so it's not going to want to take a proton, because then it would be using the electron in order to take a proton, it would have to use the electrons that are currently involved in resonance. So it would have to give up that stabilization. And so it's not going to want to do that. And so this is going to be very happy as it is without taking a proton. And so that's why you're not going to find this protonated, but you will find this protonated. In order to explain these reactions, we need to kind of get on the same base about some of these different terms I'm going to use. So these asparagine synthetase and asparaginase. These are both enzymes. So enzymes are reaction helpers, speeder uppers. They do things like help hold the reactants in the right position to react, and help the reactions happen. And what's cool about enzymes is that they speed up the reactions, but they don't get used up in the process. So they can do this over and over and over again. So you add a little bit of the asparaginase into the bloodstream, and you can deplete a lot of the asparagine and keep depleting it, even if those cells start pumping it out. And so that's an enzyme is going to speed up a reaction without getting used up in the process. Another term I'm going to talk about is nucleophile and nucleophilic attack. So basically, nucleophiles are atoms that have more electrons or electron density than they want. We're often looking at something with a lone pair, like an oxygen or a nitrogen, or a double or triple bond. These, we call them nucleophiles because they love nuclei, because nuclei are the part of the atom where the protons are located, and protons are positively charged. And so these are going to attack positively or partially positively charged things. So this includes things like the phosphorus in the phosphate groups. And so basically this phosphorus is going to have the oxygens are basically stealing the electrons from it, because the oxygens are what we call electronegative. And I have much more on this in other posts. So I'm not going to go into the details here, but basically this phosphorus is going to be partly positive, and therefore it's going to be what we call electrophilic. So an electrophile is the opposite of a nucleophile. It doesn't have enough electrons and it wants more. And so a nucleophile can attack an electrophile, and this is going to be able to do things like generate new bonds. So a phosphate, we'll see this phosphorus group be a subject to attack, as well as a carbonyl carbon. So again, oxygen is really electronegative. It hogs electrons. It's going to draw away the shared electrons from this carbon, making this electrophilic. And we'll see attacks at both the phosphate, at both the phosphorus and at a carbonyl carbon. And when you get these attacks, you can have a nucleophilic substitution reaction, where basically the nucleophile attacks the electrophile, and then you have something get kicked off, and that something that gets kicked off is going to be your leaving group. And so let's talk about this. So those are the basic concepts and types of reactions, and now let's look at how they actually go down. So we're going to take this aspartate and we're going to transfer an amino group from glutamine. So we're going to take this end group off of glutamine and stick it onto aspartate. This is going to give us asparagine and glutamate. But we can't just straight up take this nitrogen group and just like swap it with this aspartate. Instead, we need to make it so that these molecules are going to like want to react with one another. And the way that we're going to do this is we're going to have to spend a little energy money. And so when we talk about energy money in biochemistry, we're typically talking about ATP, adenosine triphosphate. So triphosphate, basically you have these three negatively charged phosphate groups that are held together, and opposite charges charges repel each other. So this is kind of like holding together a clamp spring. In order to hold these negatively charged groups together, this is going to be what we call a high energy bond. If we break it, then we can release that energy, and we can use it to do other things like get unfavorable reactions to happen. And so if you have a reaction, there's often going to be like a transition state, something that's going to make it harder to get to the other side of the reaction. And you can use the energy from breaking up ATP in order to help get over the hump. And so it's kind of like if you're sitting on a really uncomfy chair and there's a comfy couch on the other side of the room, you might really want to go to that comfy couch, but that requires an activation energy. You actually have to get up and go. And so you can use energy from ATP in order to help make these reactions happen that will make the molecules happier overall in the end. But initially you need to kind of convince them that they want to go. And so ATP is going to be able to help do this. And so what happens in the case of this is that you're having ATP kind of activate the aspartate. So we're going to be taking this aspartate and we're going to be attacking that phosphorase that we talked about, which is vulnerable to nucleophilic attack. And what happens is we're going to kick off this pyrophosphate. So there's two phosphate groups, and we're going to leave us with this AMP. So we get this aspartyl AMP intermediate, so adenosine monophosphate, where you have these two phosphate, or you have this one phosphate group left over. And then this is going to be attached onto the end of this aspartate. So now instead of just an oxygen here, we have this AMP. Why does this matter? Well, if we think about what's going to happen next is we're going to have a nucleophilic attack of that carbonyl carbon in order to kick off the AMP. We're going to attack that with the nitrogen, or ammonia group, which we're going to see how we get in a second. But basically what's going to happen is we need to make this a better leaving group. So if we just have an oxygen, that's a really, really bad leaving group. So remember something, when we attack the nucleophile, attacks the electrophile, now you can have too many bonds formed. And so something gets kicked off, and that thing that gets kicked off is the leaving group. Now, the happier the leaving group is on its own, the more willing it is to leave. And so the more likely this reaction is to happen. But if you just have a really bad leaving group, like an oxygen, that's going to not want to happen. So an ammonia wouldn't just attack this carbon as is. But if you make it a better leaving group, if we add this AMP, well, now this AMP, this will be happy by itself. And so it'll be happy to be kicked off. What's also going to be important is that, remember, oxygens are going to pull electrons away. And so what's going to happen is that this oxygen is going to pull electrons away from this carbonyl carbon. And this is going to make this carbonyl carbon more electrophilic. And so it's going to be more vulnerable to attack. And so we can attack it with ammonia. But where are we going to get this ammonia? Well, we want to get the ammonia from the glutamine. And so the glutamine, remember, it has an extra amino group compared to glutamate. And so it has an amide instead of a carboxylate. And so if we remove that amino group, well, now we can take it and add it. So what happens is this is asparagine synthetase. It actually has two active sites. So two places on the enzyme where the reaction is going to happen. At one site, you're going to have this aspartate get activated. And then nearby, you're going to have the glutamine get deaminated. And this is going to be important because, well, now you have the ammonia made like right where it is, and you can kind of just channel it through the protein in order to attack. So normally we try to avoid making ammonia because this can this can build up and cause problems. So we have like these controlled ways that we react it or that we produce it. And in this case, we're kind of producing it right where we're going to use it. And so this happens in one side on the enzyme, and this happens on the other side of the enzyme. So the water is going to help with, so a hydrolytic reaction, you're going to use water to help break this amino group off. And then you're going to take it as ammonia, and this is going to attack that carbonyl carbon. And so this nitrogen, it's going to have a lone pair of electrons. It can act as a nucleophile, attack this electrophile, kick off the AMP. And this is going to give you asparagine, which is what we were trying to make, right? Okay, so we've done the first step. We've made the asparagine. But now what about the second step? The second step is going to be breaking down the asparagine. And here we're going to see another nucleophilic attack. In this case, we're going to have the attack coming from a catalytic threonine on asparaginase. So basically, threonine is another one of the amino acids, and it's nucleophilic. It has a nucleophilic hydroxyl group. And so this is going to enable it to attack an electrophile. And right now we have this carbonyl carbon is able to act as an electrophile because it has the electrons being drawn away from it from the oxygen and from the nitrogen. And so this carbonyl carbon is going to be vulnerable to attack. And so we can have this threonine of the enzyme attack. So here we're seeing things attacking that weren't actually part of the enzyme. But here, this is actually a part of the enzyme. So we're going to get stuck onto the enzyme for a minute, and then we're going to get broken off the enzyme. But the enzyme is actually going to attack asparagine and get it temporarily stuck. This is going to release ammonia, and you also get what we call a beta-acyl enzyme intermediate. So an acyl group, an acyl is basically where you have a carbonyl carbon, and it's going to be attached to a carbon on one side and something else on the other side. And that something else could be a carbon, and it could be something else. And in the case of the asparagine, what we have here is we're going to have a beta-acyl intermediate. So beta, this is the alpha carbon, the carbon that's attached, like the backbone carbon that's going to be attached to the carbonyl group, or sorry, to the amino group and to the carboxyl group. And that's going to be attached to a carbon. This is the alpha carbon. If you go one out, this is the beta carbon. The beta carbon is then going to be attached to this acyl group. And so you have that where you have a carbonyl carbon, and then it's attached to a carbon on one side, and then something else. And so the something else is going to be attached to the oxygen of that threonine on asparaginase. Now what's going to happen is that you have this intermediate where this is temporarily stuck on here. So we just need to release it, and then we'll have aspartate, and we'll regenerate our asparaginase. And so water is going to come in and act as a nucleophile, attack that carbonyl carbon again. This poor guy keeps getting attacked by things. But in this case, it's going to then give you aspartate and your asparaginase regenerated. Then if you want to break this down further, you can. So right now we have to go through special things to break off this end amino group. But now this is just like any other amino acid. It's got only a single nitrogen group we need to get rid of. And so it can be gotten rid of through normal pathways like transdeamination, where basically you take that amino group, you pass it on to alpha ketoglutarate to make glutamate, and then that glutamate can get oxidatively deaminated, and basically take it off as ammonia. So this is called transdeamination, and you do it with a bunch of different amino acids. Like all the amino acids can get rid of their amino group this way. It's just when we have extra amino groups, like in the case of asparagine, that we had to go through other pathways in order to get rid of the first amino group. But the backbone amino group that they all have, this one we can go through a similar pathway where you might have some different transaminases for the different amino acids. But it's all uses a similar process, and you end up with this glutamate that can then get processed. So if you remove that amino group from the aspartate, what you can do is you can get oxaloacetate, and so oxaloacetate can be used to make glucose. So we call asparagine as well as aspartate glucogenic. Just a final note is that, as you might have guessed, the name for asparagine comes from asparagus. It was found in 1806 by Vauquelin and Robiquet, and it was actually the first amino acid to be discovered, but they didn't know that they isolated a protein letter. They weren't looking for those. They were just trying to figure out what was in asparagus juice, and they left some of it out to sit out when they went on a trip, and they came back and they found crystals of it. And it is not asparagine. Asparagine is in asparagus, but it's not what makes asparagus smell. Asparagus apparently smells because of something called asparagusic acid. And some people can't smell it because different people have different versions of the gene for different smell receptors. And so some people thankfully can't smell it. I can, and I think it smells gross. But anyway, that is asparagine, the basics of asparagine and asparaginase treatment for ALL. So basically, the levels of asparagine depend on the levels of the maker and the asparagine synthetase and the breaker, asparaginase. Normal cells make enough of the maker and enough of the breaker so that they can keep healthy levels. But in ALL, these cells don't make enough of the maker. And so what happens is that they typically rely on getting it pre-made from the bloodstream. You can add asparaginase to deplete the asparagine that was pre-made in that bloodstream. And this is going to make it so that those cancer cells can't get it pre-made and they can't make more, so they're going to be selectively killed. There are a number of different asparaginase treatments available, made from different types of bacteria, as well as different purification strategies, whether they're made whether they're made like just taking the native or the type the bacteria makes itself, or whether they're made recombinantly, so like in a lab, and then whether or not they're pegylated, so they have additional molecules added on to kind of try to hide it from the immune system and make it more soluble. I will post links to these various resources, and I hope that you found this post helpful and informative.